Thank you, Rudy. Once again, I want to welcome all of you to this open session of our council meeting, and that includes both those of you in the room, but also those who are joining us remotely by live webcast. And as a reminder, my director's report presentation is being videotaped, and that recording will be made available as a permanent archive on NHGRI's website, genome.gov. For those who are new to my director's report, I want to make you aware of the electronic resource that gets created for each of my director's reports. Analogous to supplemental materials of a published paper, this resource can be accessed at the URL shown at the bottom of the slide. The slides that I showed during my director's report are also available electronically, both in PDF and PowerPoint formats. And when documents are referred to or relevant websites associated with a particular slide are relevant, you will find a document number at the bottom right side of that slide. And that document number then references materials that can be accessed or downloaded from the web page shown here. And this entire dedicated web page and linked documents will be archived on genome.gov as a historic record of this meeting. Now, there's a number of other presentations that will take place during the open session of the council meeting. My report is tailored around those presentations, so I won't discuss in detail the topics that others will cover. Immediately after my director's report, we will hear about recent activities of the Inter-Society Coordinating Committee for Practitioner Education in Genomics, and the group's co-chair, Richard Haspiel, will give that presentation. And then after lunch, we have a busy afternoon. We have a total of 6 concept clearances that will be presented by staff of the Institute's extramural research program. Genomic community resources will be presented by Chris Wellington. And ELSI program announcements for unsolicited applications will be presented by Joy Boyer. And then after that, 2 concepts related to genomic technology development will be presented by Mike Smith. Mendelian genomics, solving the unsolved, will be presented by Lisa Chadwick. And then finally, polygenic risk scores from populations of diverse ancestry will be presented by Lucia Hendorf. So for the rest of my director's report, I will follow an outline that I've used now for many years that works well. These 7 areas provide a nice framework for reviewing the topics that I think people want to hear about, starting with some general NHGRI updates. As a reminder, NHGRI partners with the American Society of Human Genetics, or ASHG, in sponsoring 2 fellowships, the Genetics and Public Policy Fellowship launched in 2002, and the Genetics and Education Fellowship launched in 2014. The Genetics and Public Policy Fellowship provides each fellow with an opportunity to gain policy experience at NHGRI, at ASHG, and in the US Congress. And this year's fellow will be Samata Kata. Samata received her PhD in neuroscience from Stanford University earlier this year. The second fellowship program, the Genetics and Education Fellowship, provides each fellow an opportunity to conduct educational research and develop educational programs for a wide range of audiences, both at ASHG and working at NHGRI. The fellow also has the option to do a rotation with a public or private education organization. And this year's fellow will be Sarah Robbins, and Sarah just completed her PhD at Johns Hopkins University. Another partnership, NIH partners with the American College of Medical Genetics and Genomics, or ACMG, to sponsor the NIH ACMG Fellowship in Genomic Medicine Program Management. Now, this fellowship involves a collaboration of 4 NIH entities, NHGRI, the National Heart, Lung, and Blood Institute, or NHLBI, and the National Institute of Minority Health and Health Disparities, or NIMHD, and then also the All of Us program. Now, the goal of the fellowship is to increase the pool of physicians trained in managing research and implementation programs in genomic medicine. And up to 2 qualified physicians are selected annually and provided the opportunity to acquire credentials and experience in leading genomic medicine research and implementation programs at the NIH and at major medical centers and other organizations. Applications for the 2-year fellowship are due annually on December 1st. This year's fellows are Marie-Louise Brennan, a medical geneticist trained in the Stanford Clinical Genetics Program, and Huada Hanish, a cardiologist and echocardiographer from NHLBI's Intramural Research Program. We welcome both of them to this program. The Genomics and Health Disparity Lecture Series is wrapping up its 2019 series. This year, we had 2 excellent lectures by Manola Pereira of Northwestern University's Feinberg School of Medicine and by Katrina Armstrong of the Massachusetts General Hospital. And then in December, Ali Gharavi of Columbia University will present. Now, each of these speakers brings a unique perspective on how the field of genomics can play a part in improving health equity and continue to be mindful about the role of emerging technologies in health disparities. The talks cover a range of topics ranging from improving the diversity in the populations found in genomic databases to ensuring equitable access to genomic medicine. Importantly, each of these lectures and previous ones in the series are available online at NHGRI's Genome TV channel of YouTube. In August, The Lancet published a series of 5 review articles focusing on genomic medicine implementation. Terry Manolio, director of NHGRI's Division of Genomic Medicine, led the efforts of the Genomic Medicine Working Group of this council to identify critical topics and produce the 5 papers. She also spearheaded one of these reviews describing the major technologies to measure genomic variation that are currently of clinical value. And on top of that, Terry is also profiled in one of the articles that accompany the review papers. Now, the other 4 reviews discuss pharmacogenomics, drug safety and efficacy, the use of genomic medicine for undiagnosed diseases, using family health history to assess risk, and assessing clinical outcomes and amassing evidence to better incorporate genomic techniques in clinical care. Now, NHGRI's Genomic 2020 strategic planning process continues to move forward en route to developing our new strategic plan, which will be published in October of 2020. Recent events have included a community town hall at NIH, 2 ELSI strategic planning webinars, a town hall at Massachusetts General Hospital, and a workshop on perspectives in comparative genomics and evolution. And in terms of this last workshop, the objective of it was to review the current state of comparative genomics with a focus on commonalities across genomic investigations into humans, model organisms, agricultural species, wildlife species, and microbes. And a video cast of that workshop is available online on our Genome TV channel of YouTube. And then lastly, with respect to strategic planning, later this month, NHGRI will hold a strategic planning workshop on genomics in medicine and health that aims to explore what is needed to integrate genomics into routine medical practice in a way that prevents disease and improves the health of all members of diverse communities. And lastly, I recently discussed some of the key aspects of our Genomics 2020 strategic planning process in a Q&A that is available on the American Society of Human Genetics website. This feature captured some of my reflections on the differences between this round of NHGRI strategic planning and previous ones, how NHGRI should position itself at the forefront of genomics, and how NHGRI and ASHG can partner in areas of common interest. Moving on then to a series of general NIH updates. In June, former NIH Director Jim Weingartner passed away. Jim served as NIH Director from 1982 to 1989. And as NIH Director, he handled the biomedical research response to the HIV/AIDS epidemic and the emergence of recombinant DNA and other ethically charged biotechnologies. But of particular relevance to NHGRI, NIH's leadership in the Human Genome Project began under his guidance. He was also instrumental in setting up the Children's Inn at NIH, which is a residential facility that allows families with seriously ill children participating in research at NIH to stay free of charge. And last month, Donald Lindberg, former director of the National Library of Medicine, or NLM, passed away. He was one of the longest-serving leaders at NIH, having served as NLM director for over 30 years. Don was a pioneer in applying computer technology to biomedical research and health information. Under his leadership, NLM embraced the internet, developing and improving access to research and medical information through PubMed and PubMed Central and clinicaltrials.gov and Medline Plus, etc. He played an integral role in establishing the National Center for Biotechnology Information, otherwise known as NCBI, which provides worldwide access for biomedical information and genomic data. In terms of leadership transitions, in July, Paul Sieving retired as the director of the National Eye Institute, or NEI. Paul served in this capacity for almost 20 years, during which he established major clinical programs aiming at preventing and treating eye diseases. And then as a scientist, and through his own intramural research efforts, his laboratory developed the biological understanding of X-linked retinoschisis, a human condition leading to retinal degeneration, and then further went on and initiated the first ever human gene therapy for this disorder at the NIH Clinical Center. Following his departure from NIH, Paul will launch and direct a new Center for Ocular Regenerative Therapy at the University of California, Davis. And then Santa Tuminia will serve as the acting director of NEI while the search for a permanent director is conducted. Another transition, next month, Linda Birnbaum will retire as the director of the National Institute of Environmental Health Sciences, or NIEHS. She served in this capacity for over 10 years and as a federal scientist for nearly 40 years. Linda is the first board-certified toxicologist and the first woman to ever serve as NIEHS director. Under her leadership, NIEHS became a world leader in toxicology and environmental health research, with NIEHS science inspiring health policy and safety standards in the United States and abroad. Richard Wojcik will serve as the acting director of NIEHS while the search for a permanent director is conducted. And then last week, Francis Collins announced that the selection of Susan Gregaric as the NIH Associate Director for Data Science and the Director of the NIH Office of Data Science Strategy, replacing Phil Bourne, who departed NIH in 2017. Now, since 2013, Susan has been the director of the Division of Biophysics, Biomedical Technology, and Computational Biosciences at the National Institute of General Medical Sciences. She also served as a senior advisor to the Office of Data Science Strategy since November of 2018. And in this new role, Susan will lead implementation of the NIH Strategic Plan for Data Science, which was released in June 2018 and aims to modernize the NIH-funded biomedical data science ecosystem. And finally, of great interest to everyone, here's some interim update information regarding the NIH budget situation for next fiscal year, in other words, fiscal year 2020, which begins on October 1st. Well, with only 2 weeks left before the new fiscal year, much remains to be done. But here's where things stand as of now. So shown in the second column from the left are some relevant numbers for the Labor, Health, and Human Services Appropriations Bill for this fiscal year, in other words, 2019. And then in the third and fourth columns, the numbers passed by the House for fiscal year 2020. As you can see, the House has proposed giving NIH and NHGRI a 5% increase next fiscal year. Meanwhile, the Senate is behind in this process, but the word is that they are inclined to give NIH an even larger increase than the House, perhaps as much as 7.7% increase, although the specific amount intended for NHGRI is not yet known. Well, needless to say, all of this is still quite fluid for many reasons, many, not most, political, and we face the very real possibility of a short-term continuing resolution, or CR, on October 1st to give additional time for all these budgetary details to be worked out. But if things end up landing where both the House and Senate seem to be heading, NIH and NHGRI would end up with a healthy and much appreciated budget increase for fiscal year 2020. So obviously, stay tuned. Moving on to some general genomics updates. The research community recently lost a key contributor to the genomics revolution. Kary Mullis, inventor of the polymerase chain reaction, or PCR, and Nobel Prize winner in 1993, passed away last month. The availability of PCR was absolutely instrumental to the success of the Human Genome Project, with PCR-based methods now being central to myriad genomic applications. In July, the White House awarded the latest set of Presidential Early Career Award for Scientists and Engineers, or PCAS. The PCAS is the highest honor given by the U.S. government to outstanding scientists and engineers who are beginning their independent research careers and who show exceptional promise for leadership in science and technology. This year, 3 of the awardees are funded by NHGRI. Extramural research Neville Sanjani of New York University and the New York Genome Center received the award for his pioneering approaches to the study of transcriptional regulation. Adam Filippi from NHGRI's Intramural research program received the award for his seminal work on single molecule DNA sequencing, which was producing the assembly of complete genome sequences. And David Zhang of Rice University received the award for important and inventive research on the enrichment of DNA and RNA sequences based on rapid and highly specific hybridization kinetics approaches. A good colleague of NHGRI's and Genomics community, Peter Donnelly, who currently serves as the Chief Executive of Genomics PLC and Professor of Statistical Science at Oxford University, has been awarded a knighthood in the United Kingdom for his services to the understanding of genetics and human disease. Congratulations to Peter. And then, of course, moving on to genomes in the news. As always, there have been a number of recently generated genome sequences reported since the last council meeting, which wasn't that many months ago, but here is the list. The almond, the Siberian hamster, the avocado, the Komodo dragon, the white-footed mouse, the zebra mussel, a total of 44 ruminants, cocado, huya, the pea, the cowpea, everyone's favorite, the bed bug, and 101 genomes from the Vertebrate Genomes Project. Now, some of the genomes included in the 101 Vertebrate Genomes Project represent genome sequences that had previously been assembled. And to be honest with you, Chris Wetterstrand didn't go through and take the time to figure out which of these species represents newly sequenced genomes. But there is one worth calling out, the vaquita, and this porpoise is very cute, but sadly is on the brink of extinction. So, moving on then to the NHGRI extramural research program. And as often the case, we'll start with our genome sequencing program, or GSP. The GSP aims to use genome sequencing to identify genes and genomic variants underlying human inherited diseases. Now, one of GSP's components is the Centers for Common Disease Genomics, or CCDGs, which aim to discover the genomic architecture of common complex diseases. To date, the CCDGs have sequenced over 91,000 whole genome sequences and 105,000 exome sequences across 3 disease areas: cardiovascular, immune-mediated, and neuropsychiatric. The centers project that they will generate approximately 141,000 whole genome sequences and 212,000 exome sequences by the end of 2020. And NHGRI recently provided one of those centers, specifically the New York Genome Centers, to resequence the canonical 2,504 samples from the 1,000 Genomes collection at significantly higher quality. The resulting data are now available in an open access fashion, that is, with no data use limitations, by several means, including a Google Bucket, a workspace in the NHGRI-sponsored Anvil platform, and the International Genome Sample Resource, or IGSR. Another component of our GSP is the Centers for Mendelian Genomics, or CMGs. And to date, the CMGs have obtained samples from over 6,100 participants and have identified many variants implicated in Mendelian disorders. Using conservative, or Tier 1 criteria, the CMGs have identified 3,048 candidate variants, 40% of which are novel discoveries. They have found an additional 1,595 candidate variants that meet Tier 2, or suggestive criteria, 97% of which are novel discoveries. Now, the CMGs are committed to sharing these findings with the broader research community, and they do this in multiple ways. First, the CMGs have submitted over 125,000 entries to the Matchmaker Exchange Network, which is a federated platform that helps researchers, clinicians, families, and participants identify matches of rare disease patients with similar genotypes and phenotypes. Additionally, the CMGs contribute variant-level information to Variant Matcher, a matching database created by the Baylor Hopkins CMG, and also to ClinVar, an aggregate database for variants of clinical significance. There are CMG data currently in dbGaP, and the groups are also working to deposit sequence-level data to the Anvil platform. Finally, the CMGs have published over 623 papers since the program's inception in 2012. The GSP analysis centers have to date published 57 papers, 2 of these published since the May 2019 council meeting that warrant specific highlighting. Wang et al. Reported their integrative risk gene selector, or IRIGS, method, which can be used to integrate multi-omic data and gene network information to identify genes that may confer disease risk at known genome-wide association study loci. The authors tested their method on schizophrenia GWAS loci and identified high-confidence risk genes with significantly enriched heritability. Additionally, the risk genes identified are good candidates for drug therapeutics. Zhu et al. Identified 2 genomic variants associated with the specific Epstein-Barr viral subtypes that are also associated with nasopharyngeal carcinoma. The cumulative effects of the 2 variants contribute 83% of the known risk of developing this cancer in populations from southern China. The authors hope that it will be possible to identify people at high risk for developing nasopharyngeal carcinoma in that region so they can receive preventative therapy. The Human Genome Reference Program represents NHGRI's continued investment in maintaining the Human Genome Reference Sequence. NHGRI is currently reorganizing and refocusing our contribution to this reference by establishing a multi-component program intended to enable an improved human genome reference sequence for the research community and to foster its long-term sustainability and improvement. Awards for 2 centers were recently made. A Human Genome Reference Center at Washington University will serve as the coordinating center. They will maintain and update the reference sequence, support state-of-the-art reference representations, and educate and coordinate with the research community, including clinicians and basic research scientists. A second center, a high-quality human reference genome center at the University of California, Santa Cruz, will collect additional DNA samples from populations not represented in the current reference, including the creation of cell lines. They will then generate at least 350 high-quality reference genome sequences, a subset of which will be finished telomere-to-telomere genome sequences. The center will also disseminate the data and work closely with the other Human Genome Reference Program components. NHGRI's Technology Development Program continues to develop improved technologies and innovative solutions to enable genomic discovery. The annual Advanced Genomic Technology Development meeting was held at Northeastern University in May of this year. Nucleic acid sequencing and genomic technology development grantees presented their work and engaged in several discussion sessions. Trainees were heavily involved in the meeting, organizing their own session on career development. The third day involved a public meeting that was open to the broader research community and included presentations on spatial transcriptomics, genomic medicine, and protein engineering. The public meeting also included a Genomics 2020 strategic planning session with discussions about 3 topics: nucleic acid technologies, chromatin structure and gene regulation, and functional genomics. Meanwhile, the next due date for the NHGRI Novel Genomic Technology Development Program announcement is October 2nd of this year. Now, 2 recent papers emanating from our technology development program are worth highlighting. Weinstein et al. Developed DNA microscopy, an entirely new non-optical technology to visualize nucleic acids in a cell utilizing the power of molecular biology combined with next-generation DNA sequencing and computational analysis. And Noakes et al. Revolutionized the decades-long concept of flossing a DNA strand back and forth through a nanopore. These researchers used a helicase to move the DNA through a protein nanopore base by base and a variable voltage to simultaneously move it back and forth rapidly on a sub-base pair scale. Their generalizable single-molecule biophysical approach achieved significantly increased nucleic acid sequencing accuracy, work funded by NHGRI to advance novel DNA sequencing technology development. Moving on to the Encyclopedia of DNA Elements, or ENCODE project. The goal of this program, of course, is to create catalogs of all functional elements in the human and mouse genomes and to make those catalogs available as a resource to the biomedical research community. To achieve this goal, ENCODE periodically holds meetings and workshops to aid utilization of ENCODE data for both new and experienced users. The ENCODE Consortium held its 3rd Research Applications and Users meeting in Seattle this past July with about 200 attendees. The meeting featured keynote lectures by Nancy Cox and Steve Heinekoff, along with numerous hands-on workshops that taught users how to navigate, analyze, and integrate ENCODE data. Topics for these hands-on workshops included accessing ENCODE's 15,000 plus data sets, using the GenCode reference annotations in human mouse genomes, or human and mouse genes and transcripts, and neural networks and deep learning, and also applications to genomics and gene regulation. Now, to continue such outreach efforts, the ENCODE Consortium is taking their show overseas and is planning for the next users meeting in Barcelona, Spain next summer, aiming to reach ENCODE users in Europe. The research community continues to utilize ENCODE data in new and innovative ways to better understand the human genome and its role in health and disease. In particular, computational and deep learning methods have been utilized to analyze and draw functional conclusions from ENCODE data. Highlighted here is one such method developed by Zhang and colleagues. Deep learning augmented RNA-seq analysis of transcript splicing, or DARTs, uses ENCODE and roadmap RNA-seq data to predict transcriptional alternate splicing. On the left is an overview of this computational program demonstrating the input of ENCODE and roadmap data to train DARTs to make predictions on splicing outcomes. Shown on the right are features critical for training DARTs, including both genome sequence features and RNA binding protein features. DARTs has the potential to uncover more alternately spliced transcripts than conventional RNA-seq splicing analyses, particularly for genes expressed at low levels. And this work was funded in part by the NHGRI-supported Genomics of Gene Regulation project. Our Centers for Excellence in Genomic Science, or CEGS, program supports interdisciplinary research teams to develop highly innovative approaches in genomics research. And this summer, NHGRI renewed an existing CEGS and also awarded a new one, bringing the program's total centers to 10. The Center for Personal Dynamic Regulomes is led by Howard Zhang at Stanford University. For its next phase, the center aims to improve the precision and effectiveness of disease diagnostics and treatment by making it possible to rapidly and comprehensively track the regulome, defined as all the gene regulation switches using clinical samples such as human biopsies. The Center for Multiplexed Assessment of Phenotype is a new CEGS that will be led by Stan Fields and Doug Fowler at the University of Washington. This center will develop novel technologies to generate libraries of variants in human genes at unprecedented scale and throughput, and to assess the impact of this variation on molecular and cellular processes. And just by way of information, the next annual receipt date for CEGS applications is May 18 of 2020. The Population Architecture Using Genomics in Epidemiology, or PAGE program, is expanding the understanding of ancestral differences in genomic disease associations. The program recently published a flagship paper in Nature using strategies tailored for multi-ethnic populations. What PAGE did was conduct a GWAS of 26 phenotypes and roughly 50,000 non-European individuals, confirming 1,444 associated variants and finding 27 novel loci. Now, ancestry is more complex than simple categories suggest. And this figure shows a principal component analysis of the PAGE populations, with each color denoting a different self-reported continental ancestry. Admixture is denoted by similarly colored dots showing a continuum of genetic ancestry. This spectrum is also seen within each self-reported ancestry. The paper also quantified the added value of diversity in GWAS. First, using existing data from a European ancestry height GWAS, investigators showed that analyzing these genomic variants in follow-up studies results in more variation in height explained in European ancestry, about 15%, compared to non-European ancestry populations. Adding an additional 50,000 European ancestry participants to this analysis widens the gap, while adding an additional 50,000 non-European ancestry participants narrows the gap. So these findings suggest that we risk exacerbating disparities in genetic knowledge unless diverse multi-ethnic research is conducted. The Electronic Medical Records and Genomics, or eMERGE network, conducts genomic discovery and clinical implementation research by leveraging data from large biorepositories linked to electronic medical records. Now, the eMERGE Network recently published a paper in the Journal of Biomedical Informatics describing how conversion to the Observational Medical Outcomes Partnership, or OMOP, a Common Data Model, or CDM, improves the efficiency and accuracy of generating ephenotypes in a federated electronic health record environment. Now, establishing ephenotypes requires combining a wide range of electronic variables to describe a medical condition. However, the disparate EHRs represent these variables using a different nomenclature, making it difficult to compare the results between EHRs. The results of this work highlight the value of using the OMOP data model when generating ephenotypes to expand use of federated EHR data for genomics research. The Clinical Genome Resource, or ClinGen, evaluates and disseminates the clinical relevance of genes and genomic variants for use in precision medicine and research. And ClinGen's variant curation efforts have been scaling up. 4 new variant curation panels have recently been launched, specifically those focusing on very long-chain acetyl-CoA dehydrogenase deficiency, hereditary hemorrhagic telangiectasia, FBN1 for Marfan and hereditary thoracic aortic aneurysm and dissection, coagulation factor deficiency, and cerebral creatinine deficiency syndromes. The expert curated variants and their interpretations will be available in ClinVar next year. ClinGen also recruits volunteers from the community to curate, and these efforts aim to engage, identify, and communicate with potential ClinGen volunteers and to facilitate the placement of volunteers in the ClinGen curation efforts. Via the ClinGen website, individuals can fill out a brief survey about their interests, expertise, and desired level of involvement. They are then directed to take a series of online training modules and are then assigned to a ClinGen expert curation working group. To date, 248 people have completed the volunteer survey and 116 volunteers have completed training. Moving on, the Clinical Sequencing Evidence Generating Research, or CSER program, aims to generate evidence related to the clinical utility of genome sequencing, with a major emphasis on recruiting ancestrally diverse and medically underserved populations. In June, CSER investigators, in collaboration with the NSIGHT network, published a conceptual framework to aid the genomic medicine community in developing research questions and strategies that span discovery, translation, and implementation research. The framework outlines the 4 major domains for conducting genomic medicine research, that is, contextual factors, interventions, processes, and outcomes, each with relevant subdomains and with arrows denoting relationships among them. CSER investigators use this framework to identify relevant measures to harmonize across the consortium, which are available at the CSER website. The paper also gives examples about how the framework can be adapted for chronic disease or for pharmacogenomics research. A second recent CSER paper analyzed data on the psychological outcomes related to exome and genome sequencing results disclosure across the 7 sites from the first phase of CSER. The authors looked across multiple clinical sites and found no clinically significant psychological harms from returning these results, but there were minor differences among the sites. They suggest that future research consider the reasons behind the variation in test-related psychological responses. The Newborn Sequencing in Genomic Medicine and Public Health, or otherwise known as the ENCyclopedia program, which recall was funded by NHGRI and the Eunice Kennedy Shriver National Institute of Child Health and Health Development, has been exploring in a limited but deliberate manner the implications, challenges, and opportunities associated with the possible use of genome sequence information for the care of newborns. With the program coming to an end, the ENCyclopedia investigators presented their findings in a June public webinar featured on NPR's Mourning edition. The investigators discussed the use of genome sequencing for population screening, parental decision-making about exome sequencing, a case for preventative genomics, and genomic medicine for infants in intensive care units. Overall, the projects showed that in the public health context, genome sequencing cannot currently replicate newborn screening results, though it may be able to augment them. In the genomic medicine setting, it's particular for sick infants in the neonatal intensive care unit, genome sequencing was found to yield diagnoses and changes in medical management. And videos from the webinar can be found on the ENCyclopedia webpage. The Clinical Pharmacogenetics Implementation Consortium, or CPIC, was established in 2015. CPIC aims to provide resources needed to translate raw genetic test information into prescribing recommendations for specific gene-drug pairs by the creation, curation, and dissemination of peer-reviewed, evidence-based, freely available clinical practice gene-drug guidelines. Now, this map shows the distribution of CPIC members around the world. In June, CPIC held a meeting at St. Jude Children's Hospital to identify practical considerations for choosing a genetic testing platform and clinical laboratory, to summarize the risks and benefits of returning test results to patients and provide examples of good practices, to describe barriers to implementation of pharmacogenetics using different EHRs and strategies to overcome them, and finally, to define successful implementation of pharmacogenetics and associated improvements in patient outcome. 217 members of the consortium from 12 countries attended the meeting. Of these, 48% were from academic institutions, 10% were from healthcare systems, and 31% were from companies, including testing labs, EHR vendors, genomics interpretation services, and pharmaceutical companies. 10 days after the meeting, CPIC published a paper on Genome Web entitled Clinical Pharmacogenomic Consortium Taking Aim at Non-Actionable Genes, Countering Inappropriate Marketing. The Ethical, Legal, and Social Implications, or ELSI research program, supports research that anticipates, explores, and addresses implications of genomics for individuals, families, and communities. The program's new Center for ELSI Resource and Analysis, or CERA, grant was recently awarded to co-PIs Mildred Cho at Stanford University and Sandra Lee at Columbia University. The goals of the new center include creating a platform for resource sharing and coordination of scholarship, tools, and products, providing data curation and synthesis to identify areas of consensus, disagreement, and research gaps, and establishing a more synergistic ELSI research community by increasing availability and visibility of ELSI resources and products. Planning has begun for the 5th ELSI Congress, which will provide an opportunity for multidisciplinary ELSI researchers to gather together, share their work, and learn about other research in the field. The 5th ELSI Congress will be held in June of 2020 and will be hosted by the Division of Ethics at Columbia University in New York City. Abstracts for paper, poster, and panel presentations will be due in the late fall, and additional information about the meeting will be available soon. NHGRI offers training and career development support through a variety of mechanisms, including individual fellowship and career development awards, institutional awards, and diversity and re-entry supplements. Our programs offer opportunities at all collegiate and faculty levels. The program continues to grow with recent Diversity Action Plan, or DAP, and T32 awards. Specifically, our DAP program seeks to expose underrepresented minority students to genomic research experiences, recently having made 2 new awards. The Stanford Genomics Diversity Summer Program, led by Mike Snyder, will immerse undergraduate students in a 9-week genomics laboratory experience coupled with opportunities for professional development and mentoring. And then the Baylor College of Medicine program, led by Richard Gibbs and Deborah Murray, will make 2 tracks available for students. One track is a 10-week summer program for undergraduates, and another track is a 1-year post-baccalaureate program. All students will have the opportunity to work with mentors in developing laboratory expertise and to hone professional skills. With these new programs on board, we now have 14 DAP sites across the country. Then in terms of 232, Jordan Smoller and Heidi Rem will lead a new genomic medicine T32 training program at Massachusetts General Hospital. This award will complement the 20 currently funded NHGRI T32 programs. And our last update for our extramural program relates to the recently established Genomic Innovator Awards. These are awards that support creative, early career genomics researchers who have played key roles in consortia or similar large team science efforts. This program provides flexible research funding that allows these investigators to pursue highly innovative research in all areas of genomics relevant to the Institute's mission. And NHGRI recently awarded the first set of these grants to 6 promising investigators with the names and institutions listed here. These awards will support a number of diverse research areas, including the exploration of the non-coding genome, randomized trial of polygenic risk scoring for common diseases, and modular CRISPR genome editing technologies. And the next receipt date for this program is October 30th of this year. So moving beyond NHGRI and onto the NIH Common Fund and other trans-NIH efforts. The NIH Common Fund's Human Microbiome Project, or HMP, was active from fiscal years 2007 to 2016. The second phase of the program, known as the Integrative HMP, or IHMP, was designed to collect and integrate molecular, multi-omic properties from both the microbiome and the host in 3 longitudinal cohort studies. As a culmination of the phase 2 effort, the IHMP investigators published a collection of papers in Nature and the Nature family of journals in May of 2019. This included 4 flagship papers, over 20 companion papers, and a perspective. And all HMP multi-omic data from both phases, comprising over 40 terabytes of data, and associated tools and protocols, are available at the HMP Data Coordination Center. The Knockout Mouse Phenotype Project, or COMP2, will create and phenotype 3,000 strains of knockout mice using CRISPR technology between 2016 and 2021. The project is on track to meet its goals for the fall of 2019 and is part of the larger International Mouse Phenotyping Consortium, or IMPC. Now, in June, NIH organized a community workshop to evaluate COMP's program's goals and opportunities. Meeting attendees strongly endorsed the goal of prioritizing the phenotyping of null mutants for the protein coding genome. Given the high number of embryo-lethal and subviable strains, they supported generating conditional alleles in those genes. They also noted that the resource was quite valuable to the rare disease community. Finally, they encouraged increased outreach and interaction with the research community. This year's COMP2 annual meeting, which includes the panel of scientific consultants, will be held in Rockville, Maryland, later this year in November. The last day of the meeting will focus on COMP2's collaborations with various human disease gene discovery programs, such as the Centers for Mendelian Genomics, the Undiagnosed Diseases Network, the Gabriella Miller Kids First, and ClinGen. The GTEX portal is the primary site for accessing Genotype-Tissue Expression Project, or GTEX, data. The portal recently had its version 8 data release, which includes genotypes for over 200 more subjects and expression data for over 4,000 more samples. There is now SNP-CNV genotypes for about 900 subjects, RNA-seq expression data for about 17,000 samples, and allele-specific expression data for about 16,000 samples. Version 8 controlled access data are available at dbGaP. Raw sequences and all other protected data are now available on NHGRI's Genomic Data Repository, ANVIL. And version 8 public data, such as expression quantitative trait loci, or EQTLs, and expression tables, are integrated in the GTEX portal for public access. The central goal of Human Heredity and Health in Africa, or H3 Africa, is to develop a sustainable and collaborative African genomics research enterprise. Funded by the NIH Common Fund and the Wellcome Trust and the African Academy of Sciences, the consortium now supports 48 projects across 34 African countries and involves over 54,000 research participants. Next week, H3 Africa will hold its 14th consortium meeting in Accra, Ghana. Alongside scientific presentations and working group sessions, the meeting will also feature workshops focused on community engagement, scientific writing, and scientific communication. This year, ASHG and NHGRI have partnered to award travel fellowships for 25 African trainees and/or early to mid-career investigators to attend the upcoming ASHG annual meeting. These developing country awards, specifically for researchers in low and middle income African countries, are supported by NHGRI and ASHG and are being administered by the H3 Africa Coordinating Center at the University of Cape Town. This past summer, H3 Africa's 3 biorepositories were renewed for a second phase of funding. Going forward, these groups will focus on developing skills and infrastructure for supporting biomedical research on the African continent beyond the completion of H3 Africa's Common Fund support in 2022. So towards that end, the biorepositories will soon be launching their own website that will describe their services, resources, and biospecimen catalog for use by the greater research community, which is an essential step in their path towards sustainability. The Somatic Cell Genome Editing, or SCGE program, aims to develop quality tools to perform effective and safe genome editing in human patients. The program's goals are to develop improved delivery systems and genome editors, as well as in vitro and in vivo assay platforms to test safety and efficacy. The information and materials will constitute a genome editing toolkit that will be disseminated through a data coordination center. The program includes 6 components. The major focus is new delivery systems and rodent and large animal reporter strains. There are 2 projects focused on unintended biological effects, one based on tissue chip systems and the other aimed at developing in vivo tracking methods. There's also a small project supporting novel genome editors, and finally, a data coordination center. The second round of awards are now in process, and the next annual meeting for the program is planned for December of this year. Moving beyond the Common Fund, the All of Us research program aims to gather data from 1 million or more people living in the United States to accelerate research and improve health. By taking into account individual differences in lifestyle, environment, and biology, researchers will uncover paths towards delivering precision medicine. Now, the All of Us research program investigators recently published a special report in the New England Journal of Medicine. The paper details the goals and objectives of the program and the accomplishments made to date. And then in late August, All of Us awarded funds to Color, a health technology company, to establish the program's nationwide genetic counseling resource. All of Us is funding 3 centers to sequence the genomes of its 1 million participants, and Color's network of genetic counselors will help these participants understand the genomic information that they receive while in the program. And then in other All of Us news, Eric Dishman announced last month that he will be stepping down from his role as director of the All of Us research program and will be assuming a new position as the program's chief innovation officer. This new role will give Eric the opportunity to continue to bring new methods and processes for innovation based on his 25-year career in Silicon Valley. As you might expect, there is now an active search underway to identify a chief executive officer who will then lead the All of Us research program going forward after Eric Dishman transitions to his new role. So stay tuned on that. So moving on to the Institute's Division of Policy, Communications, and Education, just for a handful of updates. This summer, NHGRI had opportunities to bring genomics to Capitol Hill. Staff from our Division of Policy, Communications, and Education participated in the 2019 Capitol Hill Maker Fair and Senate Wellness Fair. At the Maker Fair, they demonstrated a variety of do-it-yourself biology experiments for an audience of individuals interested in grassroots science. And then at the Senate Wellness Fair, NHGRI educated Senate staffers on genetic health risk and family health history. Meanwhile, I had a chance to personally engage with Congress in July when I was invited to talk to congressional staff about precision medicine as part of the Coalition for Life Sciences annual briefing series. And through congressional outreach activities such as these, the Institute hopes to increase policymakers' appreciation for our field and offer our expert knowledge to those interested in legislation relating to genomics. In June, the NIH's Office of Science Policy launched a new resource celebrating Henrietta Lacks and the impact of HeLa cells on biomedical research over the past 65 years. This resource is intended to provide an interactive experience where users can learn how vital HeLa cells have been in moving biomedical research forward. Features include a timeline of significant research milestones that were enabled by the use of HeLa cells, a map corresponding to the origins of published research articles using HeLa cells from 1953 through 2018, a relational chart that shows the interconnectivity between HeLa cells and many essential areas of biomedical research, and links and information on the most highly cited papers related to HeLa cells from 1953 to 2018. Now, then in a related development, the Education and Community Involvement Branch worked with the Smithsonian's National Museum of African American History and Culture, and educators from DC, Maryland, and Virginia to help develop a series of high school lesson plans based on the life of Henrietta Lacks. Topics include racism in medicine, cervical cancer, and cancer treatments. Each lesson plan features a foreword by Jerry Lacks, who's the granddaughter of Henrietta Lacks. Released earlier this month, the lesson plans are available on a new webpage dedicated to the life of Henrietta Lacks and the contributions of HeLa cells to biomedical research that can be found on the Genome Unlocking Codes website. And speaking of Genome Unlocking Life's Code, this exhibition will make several more stops across North America as it continues to travel in 2019 and into 2020. The exhibition, opened last week at Turtle Bay Exploration Park in Redding, California, will remain until January of 2020. It will then travel for a stay at the Museum of Science and History in Jacksonville, Florida. Please continue to check the exhibition's website, follow it on social media for the most up-to-date program information. In late July and early August, NHGRI held its annual short course in genomics. This year's course was organized by the Education Community Involvement Branch and welcomed 25 faculty members who teaching at middle schools, high schools, community colleges, and tribal colleges from 16 states. These educators spent 4 days at NIH listening to lectures and learning about genomics teaching resources from leading NHGRI and NIH researchers, clinicians, and staff. Topics range from the microbiome, bioinformatics, CRISPR-Cas9, cancer genomics, ethical issues in genomics research, health disparities, among others. In addition, the director of the DC Department of Forensic Sciences spoke to the educators about the applications of forensic genomics, a topic of much interest to students. And the Smithsonian's National Museum of Natural History arranged a guided tour of their new Deep Time exhibition. On the last day of the course, short course alumni presented draft lesson plans on the microbiome. Other alumni developed lesson plans on gene editing and bioinformatics are also under development. Also happening this past summer, the Education Community Involvement Branch participated in a program for over 100 middle and high school students in collaboration with the Prince George's County Economic Development Corporation's Youth Career Connect program. This was the final year for the program, which was designed to support college readiness and workplace skills development for students. The program included lectures from NHGRI staff and our partners focusing on genomics, genomic careers, and hands-on activities. Based on the topics they learned about, students prepared group presentations focused on health disparities and family health history. And as you know, NHGRI strives to be at the forefront of efforts to engage and educate the public about genomics. So towards that end, the Institute recently submitted 4 proposals for panel discussions at the 2020 South by Southwest Conference aimed at increasing public genomic literacy. These included proposals on National DNA Day, creating a space for genomics in all classrooms, genomics in a sea of societal issues, and why am I Irish yesterday and Italian today? And in August, public voting for the panels was open, and an advisory board is currently evaluating the submitted proposals. And last, but certainly not least, I always like to give a few highlights about our intramural research program. And in June, Dan Kastner, NHGRI Scientific Director and Director of the Institute's Intramural Research Program, received yet another award, this case the Ross Prize in Molecular Medicine. This prize was established in conjunction with the Feinstein Institute for Medical Research and Molecular Medicine and is awarded to recognize biomedical scientists whose discoveries have transformed the way medicine is practiced. The 2019 Ross Prize in Molecular Medicine was awarded to Dan for the discovery of genetic and genomic strategies to elucidate the molecular basis and treatment of autoinflammatory diseases. So congratulations to Dan. And we have a transition. Next month, Max Munke, Chief of the Medical Genetics Branch and Director of the Medical Genetics and Genomic Medicine Training Program, will depart NHGRI to become the CEO of the American College of Medical Genetics and Genomics, or ACMG. Now, Max joined the NHGRI Intramural Research Program in 1997. Since 2000, Max has been the chief of the Medical Genetics Branch, making him the longest-serving branch chief in the NHGRI Intramural Research Program. His research has focused on the delineation and identification of the underlying causes of craniofacial anomalies in humans. And based on his work in this area, the most common form of craniosynostosis bears his name, Muncke syndrome. And we wish Max the best in his new pursuits in leading ACMG. Oh yeah, and speaking of Max, under Max Muncke's leadership, the 4th International Summit in Human Genetics and Genomics is currently ongoing at NIH. This month-long summit is designed to help developing nations build and expand their expertise infrastructure, systems, and technologies in genetics and genomics. The goal is to help participants understand the prevalence and basis of genetic disease in their nations and help them address the associated public health challenges. The summit is sponsored by a number of groups, NHGRI certainly, but also a collection of other NIH institutes and centers. It includes money raised by the Foundation for the NIH and also additional for-profit and non-profit partners. And information applying for the summit in 2020 will be made available at the summit's website later this year. And as always, the NHGRI's Intramural Research Program publishes productively between every meeting of this council. I always like to show a few research highlights. For example, Charles Rotimi and colleagues have reported the largest genomic study of type 2 diabetes in sub-Saharan Africans with data from more than 5,000 individuals from Nigeria, Ghana, and Kenya. Researchers confirmed known associated genomic variants and identified a novel associated gene, ZRANB3, which may influence susceptibility to the disease in non-Sub-Saharan African populations. Sean Burgess and colleagues have generated a high-quality draft genome sequence and gene annotations of a goldfish. The genome represents an essential resource for the study of the greater than 300 goldfish variants and for the understanding of genome evolution and related fish species. And lastly, Sarah Hall and colleagues conducted qualitative content analysis of public-facing websites for 25 direct-to-consumer genetic ancestry companies that offer services measuring Native American ancestry. Their conclusions describe how genetic ancestry companies promote a causal relationship between genetics and self-identity through marketing language such as discover yourself and are you Native American, and how these may affect tribal communities in the United States. And of course, before ending, I'd like to say, as always, if you're interested in receiving my monthly email update called the Genomics Landscape, you can simply go to NHGRI's website, genome.gov, and subscribe under email updates. And so that brings us to a close. Of course, I want to thank all the staff, at least 50, 60 of them, who've contributed to the slides and associated materials for this director's report. Without their help, this wouldn't happen. I can assure you of that. An additional thanks to the NHGRI Communications Group and web team for making my director's report an electronic resource. And as always, the ringleader, Chris Wetterstrand, a special thanks for pulling it all together. And shown here, she always provides a photo. In this photo, Chris is showcasing a new genomics timeline that we recently installed in the director suite in Building 31. So with that, I will stop and happy to take any questions before I turn this back over to Rudy.
Any questions for Eric? Okay, thank you, Eric.